And what you see kind of animated in the background are these adherent cells that are making a lot of these beautiful, thin connections amongst them in order to engage with their environment, but also engage in intercellular communication. And the story I'm going to tell you today is about how some basic principles that are based on protein curvature and balances between actin networks drive the differential formation of these protrusions that cells use to interact amongst themselves. So I really had the amazing opportunity to be shared and hosted two amazing groups that really kind of developed the research that I'm doing nowadays here, both from the lens of biophysics and also from the lens of biology. And so, as Dan mentioned, I was part of a biophysical group at the Institut Curie, headed by Patricia Bassero. And then I was also housed in a cell biology team headed by Chiara Zorzolo in the unit of membrane trafficking and pathogenesis at Institut Pasteur, where I really became interested in kind of how cells drive important shape changes in forming these thin intracellular connections between them. But a big question at the time was trying to understand, how do cells control like this common pool of proteins, the plasma membrane, to kind of drive a huge range of protrusions to form, both at small scales and at long scales to interact amongst them. So you're going to learn a bit of cell biology today. There's not any scattering, but I hope you're going to take away some key points about the functioning of the cell. And one of the key components for kind of building these protrusions is the actin cytoskeleton. And it's a beautiful polymer material that's built by the actin molecule. And depending on how actin is organized, the cell generates very distinct networks within it to drive particular shape transformations. One of the most notable structures that cells form are these branch or dendritic structures that are common to lamellipodia, which are thin sheet-like structures at the front of migrating cells. And this particular structure is dependent upon a protein complex known as the actin-related protein 2,3 complex, which is built up by 7 protein members that actually nucleate a daughter filament off a mother filament. And this particular protein will come back later in the talk. And so this complex really drives this beautiful arborization at the front of the cell. In addition to these branched actin networks, you also have beautiful linear networks that form when filaments come together and bundle to form tight packages that are important for generating protrusions. And I just want to motivate this particular structure because there's some beautiful old work from Tanya Sekina at the University of Pennsylvania, where she kind of really pioneered platinum replica electron microscopy, where you can see these beautiful actin filament bundles emerging from the surrounding lamellipodial network, for example. So the kinds of protrusions we were interested in are twofold, actually. One particular subset of protrusions are these finger-like structures that cells make at the front of their leading edge when they're migrating, and we call them filopodia, so essentially the finger-like structures of the cell. And their dimensions are such that they're actually highly curved structures, and that's dependent upon their width, which tends to range around 100 nanometers, but their length is an order of unit bigger, so they typically are around 5 microns here. So if you were a protein kind of situated inside these cellular protrusions, you would kind of see them as a highly curved negative structure, is what we would call in the field. And these structures have really cool dynamics. So this is a beautiful video taken by Betzig with utilizing his lattice light sheet microscope, where you can see these dorsal filopodia constantly extending and retracting. And the cell is utilizing this to kind of explore its immediate extracellular environment. So the functionality of a lot of these filopodia are involved in sensing the extracellular matrix, sensing each other, guiding migration of cells, and developing stable adhesions through integrins, for example. So characteristically, though, these structures obviously have important physiological function. And one of the big ones is that they mediate development of tissue. So for example, here, this is some really seminal work in fruit fly embryos, where you see growing filopodia that actually reach from one side of an opening to another to kind of zipper up the tissue and close it up via the functioning of these finger-like protrusions. However, these structures can actually be hijacked for pathological purposes. So filopodia and the proteins involved in their formation actually get upregulated in cancer cell invasion. And even bacteria can hijack these structures to gain entry into cells where they use the cell environment and create a replicative niche to multiply. And so one of those particular bacteria that's actually a big focus at Institut Pasteur is looking at Shigella flexneri, which causes dietary and other bowel disorders. These bacteria have a lot of adhesive proteins that recognize proteins localized at the tip of filopodia. And upon binding, the filopodia actually retracts, bringing the bacterial cargo to the cell where it then enters and is able to replicate and use the cellular machinery for its own development and replication into more bacteria. But filopodia at these short scales are not the only kind of protrusions that cells make. Cells also make another class of protrusions that are at longer distances. These are called tunneling nanotubes. They're similar to filopodia in that they have similar dimensions. So they're thin, roughly a couple hundred nanometers in diameter. But somehow the cell is able to tune the protein soup into making these structures much, much longer than a typical sensory filopodia. So you can see here in this micrograph image, these tunneling nanotubes can be several cell diameters in length and even reaching lengths of around 100 microns. And these structures are really important for mediating intracellular cargo transfer. So research has actually shown that these structures form open-ended conduits at certain points of time to allow the passage of molecular cargo from one cell into another. And the type of cargo that can actually be transported is really, really diverse. So not only can you have small ions like calcium, which is an important signaling molecule in cells, be transferred through these open-ended connections, but you can also have signaling RNA, proteins, and even more awesome are these transfer of organelles, such as mitochondria or lysosomes, for example. And let me replay this video on the left here, where you can see the trafficking of these red labeled mitochondria in a tunneling nanotube that are moving from a donor cell on the left, moving through this tunneling nanotube, and then entering a recipient cell on the left, or on the right, sorry. So these tunneling nanotubes are really important kind of mediating cell-to-cell communication, particularly in development, and also in mediating immune responses. Just like filopodia, though, cellular protrusions are hijacked by pathological processes. And one of the big thrusts in the Zorzalo lab at Institut Pasteur was to look at how these structures mediate the passage of aggregated or misfolded proteins like alpha-synuclein, tau, and A-beta, and trying to understand like how these proteins may be spreading in the brains of afflicted patients with neurodegenerative diseases. And for example, what you're seeing here in this video is actually a lysosome filled with the scrapie prion protein, the pathogenic version of prion, moving within these tunneling nanotubes and leaving a donor cell and entering a recipient cell. So this is kind of a cell biology understanding of how potentially these proteins could be moving about in the brain and causing pathogenesis. Aggregated proteins are not the only thing that transfer into tunneling nanotubes. We have recent evidence that viruses also hijack these tunneling nanotubes. And we've seen, for example, SARS-CoV-2 is able to move within these tunneling nanotubes and are able to infect cells that actually lack the ACE2 receptor. So non-permissive cells can actually get infected via SARS viruses that are traveling within these TNTs. But a big question is like, okay, how are these proteins or these protrusions being shaped? How is the cell kind of organizing proteins in space and time to kind of drive these shape transformations? And I wanted to just kind of take a quick step back. I probably don't need to motivate this too much, given a lot of us work with membranes, but I wanted to motivate that membranes are deformable. And so the proteins that are used to build these protrusions depend at some level on the deformability of membranes. And thanks to really some seminal work by Helfrich back in the 70s, we obviously can think about membranes and the deformability as kind of a two-dimensional sheet. And the reason being is that cells are often much, much bigger than the 5 nanometer thickness that bilayers tend to average about. So if we kind of think at a very basic level at what energy terms are necessary in describing deformability of membranes. We obviously can think about stretching the membrane and increasing its area through kind of a strain here. This obviously affects the tension, just like you would when you're blowing up a birthday balloon, right? The more air you pump into a balloon, the more it's taut, right? So this relationship between how taut it is, the tension of the membrane, and how much deformation you're doing via strain is related to a proportionality constant, which is the stretching constant. But in reality, the stretching constant is really, really high for membrane systems, and it's around 250 millinewton per meter. And so if you're actually to do controlled experiments like what Evan Evans did back in the day with micropipette aspiration on vesicles, you kind of find that you have a very small limit with which you can strain your vesicle system before the whole system collapses and ruptures. So in essence, your membranes are not super stretchy for deformable actions. What is key is actually bending of the membrane. So doing deformations in 3 dimensions here. And this is applying essentially localized curvature events in a nutshell to the membrane in 3 dimensions. And just to kind of orient ourselves, if we're a protein and we're sitting inside a cell membrane and we're looking at a deformation that's coming within the membrane towards the inside, we call this a positive deformation. So this is kind of deformations like endocytosis, for example, when cells are trying to bring in material for metabolism, let's say. And for protrusion-mediated events, where we're thinking about deformations that are extending outwards to the cell, this is something we call negative curvature. And obviously, the scale with which these deformations are occurring are dependent upon the size with which you're making these deformations. And so we can define curvature as something as 1 over the radius of a circle ascribing the deformation that we're making. And similar to stretching, there's obviously a relationship that's relating how much we're deforming to this energy of bending, and this parameter is this bending rigidity, or kappa. And what's really amazing is that based on the lipid compositions, most of these bending rigidities are just a few tens of kBt. So it doesn't take much work by proteins to apply several tens of piconewtons to make these systems very pliable or deform them. And so the protein that's really key, as I mentioned from the beginning, in doing this deformation is actin here. And just to orient you, actin has this quaternary structure of 4 subunits and a nucleotide binding clasp for ATP. And because of this quaternary structure, it actually forms polar filaments, where you form a plus-ended side of a growing filament that tends to be where polymerization occurs. And you have a minus side of the filament tending where depolymerization occurs. And so cells utilize this kind of cascade or this kind of process between polymerization and depolymerization to eventually apply localized forces to bend the membrane. And so for a basic understanding of this actin polymerization is that you can take these actin monomers that are bound with adenosine triphosphate. These get funneled to the growing plus end of the filament, often mediated by polymerases, or AKA elongation factors, like this particular protein that I'll mention here later called VASP. And as ATP hydrolyzes to adenosine diphosphate, the actin filaments age over time. And other classes of proteins recognize this aged filament and begin to depolymerize it through severing of the filament. And obviously, because it's a cycle, we can regenerate the monomers by exchanging ADP for ATP and also rebinding the monomer with monomer binding proteins that help facilitate repolymerization. So you can imagine that we have deformability of the membrane via bending. We can have localized forces from active polymerization. If you orient these polymerization processes near the membrane interface, you have a potential scenario for directed pushing forces to do this work in generation of protrusions. What's really unclear where I came in at this time was trying to understand how the actin is locally recruited. How is the cell controlling the local polymerization of the actin in generating a protrusion? So we want to know what's the spatial temporal control of the protrusion. And then ideally, once you generate these protrusions, how do you span different length scales? How do you go from a sensory filopodia up to a long intracellular tunneling nanotube that matures for cargo transport between cells? So I'm going to start briefly how we address this first topic using some really cool in vitro techniques here. And one of the key hypotheses we had is that maybe a class of proteins as kind of like the key crutch in generating these protrusions. And we thought the bar family of proteins, which is this bin Amphiphysin RVS class of proteins, particularly those that drive outward deformations like IRSP53, would be critical in generating protrusions. And so as a general perspective on these proteins, they all have kind of a typical banana-like shape where they dimerize together to form kind of a gently curved surface where they can interact with the membrane inner leaflet. They have extensive regions of organized positive residues like lysines and arginines that potentially can interact with negatively charged lipids in the inner leaflet of the plasma membrane, particularly things like phosphatidylserine or phosphoinositides, for example. And what we found previously is that this class of bar proteins can actually drive and generate deformations of the membranes at certain concentration thresholds. So it seems to be using this kind of dual nature of its intrinsic curvature to kind of impart that curvature onto the membrane to drive these outward deformations. And what's also unique about this class of proteins is that in addition to its membrane binding region, it has other domains that facilitate protein-protein binding or protein-protein friendships. And particularly this SH3 domain recognizes and binds actin modulators, particularly polymerases like VASP here. So we kind of have almost a perfect system to kind of interface the membrane to the cytoskeleton for protrusion generation. And so the hypothesis we had is that particularly the key components you need to kind of drive protrusion formation is you need kind of organization of these curvature proteins. Where, for example, you could have an initial organization or kind of clustering, you could say, of specific phospholipids that are important for recruiting IRSP53 to the membrane. And so once you have enough IRSP53, at the membrane, it can drive the deformation that's dependent upon its intrinsic curvature. And this drives deformation outward towards the extracellular environment. And because of those protein-protein interaction modules that are on the protein, it can then drive the formation of the supporting actin structure underneath this little nascent protrusion that you formed. And so as the structure matures, you then elongate and kind of form a mature filopodia, for example. So how can we investigate this kind of in a controlled manner? I mean, the cell is a beautiful system, but it's very complex with all the different regulatory processes that are occurring within it. So with one of my amazing friends and colleague, Fong, we worked to develop a reconstitution assay where we take synthetic vesicles, particularly giant unilamellar vesicles, which we call GUV, and interface them with purified protein components. So we interface it with our curvature protein, IRSP53, we interface it with our polymerase, VASP, and we interface it with monomeric actin, with the hopes of seeing these proteins come together and assemble and form synthetic filopodia that will generate protrusions towards the interior of the GUV, because we're adding these proteins to the exterior of the GUV for simplicity. So what we found is that when you simply take IRSP53, our curvature protein, and incubate it with phosphoinositides, particularly PI45P2, so it has these two phosphates, we find that IRSP53 has this innate ability to cluster and form these hotspots of high density protein patches. And I hope you can see that here in this fluorescent micrograph, and additionally in this heat map here. And depending on kind of the local density that you have, we even were able to pick up instances of tubules being formed via hot patches of these proteins here. So we see that this PIP2 lipid is kind of organizing IRSP53 into kind of localized platforms. And this behavior is completely removed simply by changing the negative charge of the lipid species. So when we switch out PIP2 and we put in phosphatidylserine of an equal molar amount, we find a complete loss or almost a complete loss of IRSP53's ability to cluster. We can quantify that cluster size, and we do see a fairly significant decrease in the cluster sizes of this protein when we measure it by confocal microscopy. So it really indicates that kind of the lipid identity is important in organizing the protein on the membrane surface prior to any kind of protrusion being formed. We then wanted to ask the question, once you have IRSP53 localized, what is then the stepwise recruitment of the polymerase of VASP? And does VASP even bind the membrane on its own? So we did this control experiment first where we take the GUV where we know it binds IRSP53. So we take our PIP2 GUV, but we remove IRSP53 from the mixture. And then we incubate this GUV with our polymerase, our actin elongation factor, VASP here. And what we surprisingly found is that VASP has no specificity for the membrane whatsoever. So VASP itself is not localized to the membrane in the absence of IRSP53. However, once you reincorporate IRSP53 and pre-incubate it, have it on the membrane surface, we get VASP binding. And VASP actually perfectly colocalizes with the IRSP53 clusters. So IRSP53 is kind of really the key linchpin in being the first member at the membrane prior to any other protrusion important proteins coming in. And what's really amazing is once we kind of increase the complexity and then added in monomeric actin, we find that protrusions generate from these IRSP53 hotspots. So we get the formation of actin filaments building these artificial filopodia out of these purified protein complexes here. And the polymerization is not dependent on the innate polymerization of the actin monomers into filaments. It's completely dependent upon the polymerase that we have. So if you remove VASP from this mixture, you don't get any tubules that are filled with actin, right? So you need the polymerase there to kind of drive the filament formation within these protrusions. So I just want to have a quick checkpoint here because maybe a lot of information. So we kind of showed that through reconstitution experiments, simple mixtures of PI45P2 is alone to self-assemble these proteins into clusters, and that these clusters localize key actin polymerases to the membrane in a kind of ordered fashion. And that once you have VASP, it generates the actin filaments necessary for supporting the protrusion. But this kind of lets us kind of begging the question in the sense like, if IRSP53 is such a prone initiator of protrusions, it's essentially like ready to go make these protrusions in cells, you would think the cell would want to have a control mechanism in place to kind of maybe tone down the activity of IRSP53, because you imagine cells want to be forming these protrusions in space and in time when it wants to, maybe in particular into, chemogradient cues that are going on in the extracellular environment. And so we wanted to kind of test, is IRSP53 regulated in cells for protrusion formation? And is its regulation important in driving actin filament formation? So what I developed was what I think is a pretty elegant technique where you can actually make artificial protrusions and see what kind of protein components enter these bare membrane tubes. The question is, how do you make these membrane tubes? Well, we used a micromanipulation technique centered on tightly focused laser traps where we can grab onto a sticky polystyrene bead around 3 microns in diameter, and then interface it with the plasma membrane of an adherent cell, and then relatively pull the bead away from the cell. You essentially rip the plasma membrane off the underlying actin cortex, and the system equilibrates to form a thin nanotube. And this nanotube, or tether that I may call it, has similar diameters to the endogenous filopodia or even the endogenous TNTs that cells make. So essentially we use it as a test tube to see what happens to certain recruitment of proteins and actin once you have this bare tube. And this is just a cool video showing this in process where the bead moves away from the cell and you can see this thin fluorescent tube here. Because we're constantly tracking the bead through video microscopy, we can relate bead displacements to forces being enacted upon on this bead. So the trap is acting like a Hookean spring. So if we calibrate our optical tweezer, we know the displacement of the bead, we can calculate piconewton level forces that are being imparted on this bead. And so, for example, we generate often these force trajectory plots where we look at the force being imparted on these beads from its equilibrium position, not being in contact with any membrane surface, to being pushed into the cell plasma membrane, and then pulling a membrane tether. Takes a little bit of energy to pull that membrane off the actin cytoskeleton, and then it eventually relaxes down to a static force that is necessary just to hold that tube between the bead and the cell. So we actually, as I mentioned, we use this as kind of a test tube scenario to kind of ask questions like, what will IRSP53 do? Will it recognize this nanotube? Will it then develop actin within this tube based on maybe some regulation within the cell? And I mentioned that this nanotube has very thin diameters, around 100 nanometers, just like filopodia or TNTs. So we can use it as a metric for gauging the curvature sensitivity of this protein, because we can use the cell body or the soma as a reference for zero curvature, measure the fluorescence intensity of the protein there, and compare it to how much protein goes into the tube, because the tube has a higher curvature, right? Curvature is 1/r. So in essence, we can measure the sorting ratio of this protein by kind of doing normalization of the amount of protein that we get in the tube compared to the amount of protein that you get in our reference plane of the cell body soma. So essentially, sorting ratios, or S's, that are greater than 1 indicate preferential enrichment of the protein in the tube, versus anything less than 1 will indicate de-enrichment or exclusion of the protein from the tube. So we began by taking some adherent fibroblasts and looking as a control, taking the membrane binding domain of IRSP53, which again has that intrinsic curvature and should be detecting this curvature that's being formed in the pooled nanotubes or in the pooled tethers. So this IBAR domain is tagged with the fluorescent reporter, this enhanced GFP, and we found indeed you get high signal counts for the IBAR domain within this tether. Right? And so this is just a heat map showing that ratio, or the sorting ratio, again, highlighting that you have very high sorting of this protein, this membrane binding domain within these tubes, and it roughly had a value of around 5, let's say. So a pretty high sorting preference within these curved geometries that we created with optical tweezers. What was kind of confounding though, and what you'll see here, is that when we just went up in complexity with the protein, so expressing the full-length protein, that now has its other domains attached to it, we saw kind of a dual behavior for this full-length protein. So with this fibroblast cell line, we actually have control over where we do the tube pulling. So we can do our measurements at the leading edge of the cell where it's actively trying to migrate or interface with its environment. And this zone actually has a lot of activity in membrane remodeling, for example, where you have active ruffling of the dorsal cell surface. And so if we actually pulled these membrane tubes from these zones of activity with the full-length protein, we saw very clear recruitment of the protein, as we did with the IBAR domain. And if you, however, do these experiments on the rear side of the cell, so near its butt, where not much is going on membrane-wise, we actually saw some interesting behavior that confounded us for some time, where we consistently saw de-enrichment of the protein. So here we see the membrane tube that's pulled from this cell, but there's no fluorescent full-length IRSP53 in this protein. And so obviously, visually, we see this difference, and this is reported in the sorting ratio, where we see any tubes that are pulled from non-active zones of the cell, we don't see any recognition of these tubes by IRSP53, versus where there is active remodeling of the plasma membrane, we see high enrichment of IRSP53 full length in these tubes. And when IRSP53 is actually enriched, we can actually use our force trajectory plots to report on actin development within these tubes. So initially, as I mentioned, when we pull these tubes, there's a static force that's necessary to hold these tubes. And so any kind of dynamics, i.e. visualized through peaks, would be indicative of actin growth within these tubes, as kind of hinted here with these magenta arrows. And how are we relating this to actin development? Well, if you think about the sense of like, if actin completely grows within these nanotubes and kind of forms a contiguous linkage from the actin that's present in the cell body, and then this actin that's growing linearly within the nanotube makes a linkage eventually to the tip of the nanotube you pulled, which is in contact with the bead, you now have a continuous system to transmit forces from the cell body to the bead. And so, for example, if the cell is doing any kind of contractility or retrograde chain, flip over of the actin filaments, because there's always kind of actin polymerization, actin depolymerization, you can see displacements of the bead back towards the cell body. And movements of the bead back towards the cell body is a positive force in our laboratory frame. So we see these kind of peaks of force increase indicative of these actin dynamics cycling on and off within these nanotubes. We can actually measure the magnitude of these forces, and we see pretty high piconewton level forces that these essentially pseudo-filopodia create. Let me get my pointer back on here. And we can see roughly on average, around 10 to 20 piconewtons. And what's really kind of cool is that these artificial tubes that we formed actually kind of mature into a functional protrusion, because the forces we're measuring on these beads are actually akin to the force response that people have measured on endogenously formed filopodia. What's contrary, though, is like if we look at the tubes that lack IRSP53 enrichment, so where we pulled these tubes on the rear end of the cell, all we measure is a static plateau, so the force necessary to hold this tube. So we actually don't see any actin within these tubes. So it's indicative that when you don't have IRSP53 present, you're also not recruiting the actin machinery within the tube to generate a functional protrusion. So we do also see that once you have IRSP53 enriched, just like in our reconstitution experiments, you can get recruitment of the polymerase. So this is just a fluorescent time-lapse movie showing a tube with IRSP53. We also see the presence of VASP coincident with the IRSP53. And then additionally, we see important bundling proteins are also enriched. And this is the protein FASN, for example. So we really see that this protein is, again, key in recruiting key machinery-free actin, and it seems to be regulated within the cell, very tightly so, where when the cell is at the front actively mediating its membrane remodeling, the protein appears to be in some sort of active conformation, where it's then allowing these protein-protein interaction modules to be free and bind with VASP. Whereas in the rear of the cell, the protein seems to be held in a tightly inactive state where it's not driving machinery recruitment. And there's some evidence here that possibly the switch between inactive to active may be dependent on local changes, again, in the membrane composition with PIP2 density, binding to certain partners that kind of open up this domain, or other cell regulators like GTPase molecules, which have known gradients within the cell, for example. But so I hope I kind of convinced you that we have kind of a good information on like the key molecules that are involved in generating these protrusions. Again, we need these curvature sensitive proteins to assemble, and they're key in then recruiting these actin machinery to drive protrusions. What I haven't touched on is how might cells regulate the length scale of these protrusions? How might you take a protrusion that's having filopodia lengths around, let's say, 5 microns? How do you go up to a protrusion that's 20 times longer, right? If we're going to be 100 microns in length, right? What's going on there? And this is where we did a lot of work with my cell biologist team at Institut Pasteur to kind of address this particular question. And I don't have time to go over all the amazing data that we did in this paper, but essentially we created some techniques to kind of screen for the formation of these long-forming TNT protrusions. And this involved creating patterned surfaces using deep UV micropatterning techniques, where you take a binary mask that allows the transmission or non-transmission of deep UV light, which then can burn an underlying polyethylene glycol layer that's a cell repellent to form adhesive islands for cell attachment, basically. And because we can control the distance between these adhesive islands, we can essentially know if we're forming filopodia versus forming longer distance tunneling nanotubes, for example. And this is just kind of a Z-stack image through such a protrusion being formed, a tunneling nanotube at a long distance here in the upper Z-stacks of this confocal image. And just focusing a little bit deeper, you can see it's substrate detached, which is pretty characteristic of long-forming structures, and it's also enriched with actin within it here. And what we found using this methodology to look at TNTs is we were able to kind of deduce phenotypic dependencies for protrusions of TNT length scales. And for example, we found that many of the cells forming TNTs, the TNTs were originating from cells not making lamellipodia. So again, at the very beginning, lamellipodia are those structures that are built by the activity of ARP2/3 that makes branched actin networks. So we really see TNTs originating from networks that are not dependent on the activity of ARP2/3. And luckily, this is something we can test in vitro because there's small molecule inhibitors of the activity of ARP2/3, which is the CK666 molecule here. So we again relied on this nanotube pooling assay here to look at the consequences of actin formation as a function of the amount of drug we're using to inhibit the ARP2/3 complex. So in this particular experiment, we take our adhesive microspheres. They're, just for reference, coated with a non-specific adhesive protein. And then we pull nanotubes from our cells, and we use a reporter for actin, which is a small molecule known as F-tractin. And this F-tractin is fused to the green fluorescent protein EGFP. So we can visualize the amount of F-tractin and hence the amount of filamentous actin that we have within these pulled nanotubes as a function of the amount of CK666 that we put into the system. And so this is a video of our mock condition where we just use the solvent vehicle, which is DMSO, to show that we don't see actually much recruitment of actin in these conditions here. So for example, you see here in magenta, we have the membrane signal here. F-tractin is here in the middle, and I'll replay this video. We don't see any actin growth within these tubes. And if we quantify the amount of actin through several repetitions, we typically see actin doesn't grow much more than 5 microns, which is actually quite surprising in the sense that's the typical filopodia length that cells make on average here. So we're kind of recapitulating kind of the average ensemble length that filopodia can make here. What's really quite striking, though, is once you inhibit the ARP2/3 complex, you get robust formation of actin filaments within these nanotubes here, as you see within the F-tractin signal. I'll play it one more time, and you can see this actin growing within this F-tractin signal. And if we quantify this, we do indeed see kind of a shift to longer length scales upon the inhibition of the ARP2/3 complex. And we see a lot of tubes that have actin reaching lengths of 15, 20, or even more compared to the vehicle control, for example. And again, we can support this data with our force. So we have kind of a combinatorial report here where we have fluorescence giving us this indication of actin formation, but our force is also telling us information of actin. So again, if we don't have actin growing along the entire length of the nanotube structure that we pulled, we don't have a continuous structure linking actin dynamics in the cell to what's going on at the tip that's attached to the bead. So again, we see plateau in the force that's representing the force necessary to hold that nanotube in place between the cell and the optical tweezer. And this contrasts in the sense, again, if we do form a contiguous structure, we should see dynamics in the force profile. And that's indeed what we see here when we inhibit this, the ARP2/3 complex with CK666. So we see a lot of dynamics of actin with these force profiles. I think I have enough time just to kind of touch on a bit of kind of the proteomics that we did in the sense of we see a lot of effect here on maybe a balance between different actin architectures in that if we inhibit the ARP2/3 complex, we tend to kind of favor maybe more of a linear actin network formation that drives protrusions to reach longer length scales. We wanted to kind of address like maybe what are some of the functional components that are downstream potentially of IRSP53 and ARP2/3 to kind of get a better global picture of what may be changing within the cell to allow protrusions of different length scales to be forming. So we relied on a proteomic approach in collaboration with the proteomic department at the Institut Pasteur, where we essentially did a modified immunoprecipitation technique where you can take agarose beads that are decorated with anti-GFP nanobodies, and these nanobodies recognize our GFP-tagged bait proteins, which can be IRSP53, for example. And when these GFP proteins are expressed in the cell, we can do treatments of our cells and then pull down the proteins under those treated or non-treated conditions, and then look at potential proteins of interest via analysis of mass spectrometry. So we use bait proteins like IRSP53, and I don't have the time to tell you about a lot of the data we did into identifying EPS8 as a co-partner in forming protrusions of long length scales. But this co-partner is known as EPS8, or epidermal growth factor receptor kinase substrate 8. And it's an actin regulatory molecule where it can either cap filament growth or it can actually bundle filaments together. And we found that IRSP53 and EPS8 have a specific interaction in protrusions of TNT length scales. And so we also used EPS8 as a bait protein in our proteomics study. And we know from past work too that EPS8 directly upregulates protrusions of longer length scales, as reported in this 2016 report, because we can measure increases of TNTs and we can measure increases in cargo being transferred between cell populations. So it's a known TNT effector. So by using these two proteins as bait proteins, we found some really interesting network changes on actin that seems to facilitate the really remarkable growth of very long actin filaments. So for example, when we looked at these bait proteins, we actually see a lot of loss of some proteins upon inhibition of the ARP23 complex. So we actually find direct interaction loss of EPS8, IRSP53 with subunits of the ARP23 complex. So there seems to be, once you inhibit the complex, you actually lose some direct protein-protein interactions, potentially freeing the EPS8 IRSP53 interaction module from branch actin networks. And we also see some reduced interactions with proteins that I mentioned at the very beginning that cause depolymerization of actin filaments within the cell. And these proteins are part of the cofilin, joscelin, advalin family that cause disassembly of actin filaments and regulators that turn on severing, which are these phosphatases. We also picked up some really interesting candidates that the Cellular lab is actually exploring now in the future, are some effector proteins that work downstream of Rho GTPases that have known effects on kinases that inactivate protein severing, actin severing proteins. And so these two proteins, CDC42, EP1, and BPA, have known activities at turning on limb kinases, which inactivate cofilin by phosphorylating it. So we seem to have kind of a really rich network being formed in how potentially actin filaments may be changed, where upon decreasing the branching activity of ARP23, we kind of relieve EPS8 and IRSP53 from these networks. They have less associations with actin depolymerizing factors, which kind of shifts the equilibrium balance to providing more filament elongation to drive protrusion outgrowth. So this is where I come to the end, where I'll just kind of summarize kind of the big global picture here, and something you can take away from today's work, in the sense that we found that protrusions are really mediated by the activity of curvature-sensitive proteins, particularly IRSP53, where the cell tends to keep it in an inactive state to prevent unwanted protrusion formation. Then upon activation, these proteins self-assemble in a lipid-dependent manner on the plasma membrane surface, whereupon they recruit specific actin polymerases, potential bundlers, monomeric actin to drive the generation of protrusions. And depending upon how you modify the balance between the density of branch actin networks compared to linear actin networks, we find this has a big impact on the length scales that protrusions can achieve. So here I'll come to the end and want to really give a big shout out to my two amazing mentors that I had a wonderful experience with in France, Patricia and Chiara. It was just an amazing experience to kind of get these two sides of science, from the physics perspective and the biological perspective, to really make some key findings in protrusion biology I want to thank my co-authors on these studies like Fong, Nina, and Savan on the TNT side, the many students that I had the pleasure to work with and train during those years, and particularly our collaborators from the proteomics side like Thibault and Mariette Pasteur, Stephanie Decroix-Curie for her help in micropatterning. And unfortunately, I didn't have time to talk about some other studies that we did in this project that involved Pekka and Greg Voth and his student at the time, Zach, in looking at kind of live cell protrusion dynamics and clustering of lipids via coarse-grained simulations. And obviously, I got to thank all our funding that supported these amazing works through that time. So we'll stop there and take any questions that we have.